Hi friends, my name is Reshma. I'm going to uh, discuss about the topic FRAP, fluorescent recovery after photo bleaching. When we come to the introduction, we all know there are proteins and lipids is present in the plasma membrane of all living organisms which show lateral and transversal movements. FRAP is a technique used to measure the dynamics of two-dimensional or three-dimensional movement of fluorescent labeled molecules within or between cells by measuring diffusion coefficient. And the principle followed by FRAP is to photo bleach the fluorescently labeled molecules in a small region of sample. Then the mobility of the fluorescently labeled molecule is evaluated from the recovery of fluorescence due to exchange of fluorescent labeled molecules from the surrounding unbleached area. In FRAP experiments, photobleached areas restricted and as technique name suggests the recovery of fluorescent back into is monitored. The main technique involved in FRAP was photobleaching. Photobleaching is an irreversible process that involves the irradiation of fluorophore with light. The result in the destruction of fluorophore and with ability to emit fluorescence. Um, so it loses both fluorophore and its ability to emit fluorescence. Recovery of fluorescence as a result of exchange of bleached fluorophores with those unbleached from surrounding area is shown in FRAP technique. Observing this rate of recovery provides important understanding of movement and interaction of intracellular molecules. This movement shows the diffusion of molecules in the plasma membrane. The lateral diffusion rates measured by following techniques or the methods or the steps involved in the FRAP are the following. Procedure. First one, mark the membrane protein of interest with a specific fluorescent marking group, and this fluorescent group is then bleached in a small area by a laser beam. The full area is not bleached, only a small portion is bleached with a laser beam, and that we can observe that this bleached area gradually retrieves its fluorescent signal not by reversal of bleaching effect but by the migration of fluorescent molecules into the bleached area from unbleached areas. This shows the evidence of lateral diffusion, and the rate of this lateral diffusion can be or the speed of protein molecules or lipid molecules which can diffuse laterally can be measured by calculating diffusion coefficient. This is done by measuring the time taken for adjacent membrane protein carrying unbleached fluorescent group to diffuse into bleached area and plotting graph. Here is the picture representation of procedure in FRAP or steps in FRAP. First, there is a fluorescently labeled proteins. These green things are the proteins, and the red minute things on the picture shows the fluorescent labeling. And when a laser beam introduced in there is an bleached area. Of course, there is bleached area as well as the unbleached portion. After some time, there is recovery. Of course, that means the unbleached fluorescent molecules traversed into bleached area and replace the bleached one, and the bleached areas molecules moves to unbleached area. So there is alternate there is bleached as well as unbleached molecule is present in the bleached area. Here is the FRAP curve. Fi initial fluorescent intensity, F0 when the signal drops or when we photo bleach with the laser beam, and Using this plot, we can calculate the mobile fraction and immobile fraction using the formula shown in picture, and a diffusion constant can be calculated by measuring the radius, and uh, the dynamics of the fluorescently labeled protein can also be determined. Here is the picture representation of pre-bleach, post-bleach and recovery. Here are the references.